sheep brain, and I ordered it with the dura mater because it's really nice to see how it's thick, and that's the purpose of the dura mater, to have a really tough, that's what dura is referring to, it's a tough layer covering the cerebrum, the whole brain. And we can take a look at the outer surface before we dive into dissecting it. And we have the cerebrum here, superiorly, posteriorly the cerebellum. And we'll see there's the dura mater here that we're going to have to cut through. And there's the brainstem. We can see medulla. We can already guess this is the pons, the anterior bulge here. That's the pons. The rest, we can see a little bit of the midbrain here. The rest is covered with this region. There's remnants of the eyeballs here. We won't go through that much, but you can see remnants of the skeletal muscles here. And there's the optic nerve. The optic nerve from each eye is going to crisscross here in the center and project to the occipital lobe in the back, posteriorly. So we're going to actually cut all that off because we want to get to the brain. So let's get that dura mater off first. And if you were doing this yourself, you would feel that it's pretty tough. So I'm cutting through the dura here. I'm going to cut both sides. And to remind you, the dura mater is made of two sublayers. The periosteal layer that is closest to the skull, ostium is referring to the bone, and the meningeal layer that is deeper, that is the one closer to the rest of the meninges. So I am peeling the whole dura here. Sometimes taking scissors is what's needed. We can actually also see here the olfactory bulb from the olfactory nerve for the sense of smell. Look at this dura here I'm peeling off. And I'm just going to cut through those nerves. Continue peeling. The meningeal layer of the dura separates from the periosteal layer and dives down between the two cerebral hemisphere, creating that falx cerebri. We see that here. Well, I'm really lucky. This is working very easily today. And also, before I take this out, I want to show you tentorium cerebelli, which is that layer of the meningeal, the dura, that goes between cerebellum in the back, and cerebrum. So it goes right between here. Tentorium forms like a tent over the cerebellum. Great. This is all off. So that's connective tissue that protects the brain. So there it is. Without the connective tissue protection, we have cerebrum. We don't really can identify the central sulcus, but the frontal lobe is here, parietal lobes, occipital lobe, posteriorly there's the cerebellum. Inferiorly, we can see now the whole brainstem with that anterior bulge here being the pons, so this would be medulla oblongata, pons, and midbrain part of the brainstem, and we're going to talk about that as well. So before we cut even more, we dissect more, let's peel the cerebellum just a little bit here and take a look here. And this is the back of the midbrain. And the back of the midbrain is a region called the tectum, or also corpora, that has corpora quadrigemina, the body of four twins, four And here they are. So just as I mentioned in lecture, there's superior colliculi here, inferior colliculi. They're not exactly the same sizes. Superior colliculi are a little bit larger in the sheep brain. And as I mentioned in lecture, superior colliculi receive sensory information from visual sensory information, like a flash, if you see a flash on the side, and inferior colliculi receive auditory sensory information, like a big bang you could hear. So I'm reminding you, superior is like your eyes are higher than your ears. That's how you can remember which one is which. And then from that region, we have neurons that are sending motor commands back out to the muscles of your neck for that reflex response of turning your head towards that sensory stimulus, either the flash or the big bang. All right, so the next dissection is going to be longitudinal. Across here, the longitudinal fissure, and I'm going to go straight down the whole brain as centered as possible. Oh, I already can see something really interesting here. I'm trying not to tear either. There we go. All right, this is so cool. And what do we see here? So I'm directing you to the ventricles. So here they are. We talked about that. The lateral ventricles. The first two ventricles in the cerebral hemispheres, they're called lateral ventricles. There's one on each side, and if you use your forceps and you go in like this, you can actually realize that it's a little cavity there. And if you look more in there, you can see something that actually has peeled off a little bit, and the rest is here, and this is called the choroid plexus. That's that shag rug I talked about in lecture. It's a network of capillaries the ependymal cells use. They take the plasma out of the capillaries, tweak the composition, and create cerebral spinal fluid that they dump back into the ventricles. So that's the choroid plexus. And it's so cool to be able to actually see it in the ventricles. So here we go. There's the other ventricle, and there's the choroid plexus in there as well. So really, it's just fun to actually see it. All right. So the brainstem, if we continue looking at the brainstem here, we have medulla, pons, midbrain. We know that's midbrain because we already identified the tectum here. So we know this is midbrain, and if we continue our journey up, that would be the diencephalon, and that specific area is actually the thalamus of the diencephalon. So that's the progression here. You can also see the gray matter and the white matter in the cerebellum. You see this nice folia here. The heart, it's hard to visualize the third ventricle if we're looking at the ventricular system. Lateral ventricles dump into the third ventricle, then the cerebral aqueduct, and that takes you to the fourth ventricle, which anatomically is located behind the cerebellum, between pons and cerebellum right here. So there's another section that I prepared so to show you that in a different view that would be better than this one. Here it is. So behind the cerebellum, between pons and cerebellum, you have this pocket. You see that cup down here? That's the fourth ventricle. So in every ventricle produces CSF and adds to the overall volume of cerebrospinal fluid. And that's the circulation. And remember, it's at the level of the fourth ventricle that you have the median and lateral apertures, little trap doors, if you remember, where the CSF escapes from that enclosed system and bathes the whole brain. Additionally, I want to show one more dissection here. If we dissect to do a cross section here of the frontal lobe, we get an actual better view of the gray and white matter in the cerebrum. So that would be the cerebral cortex, where the cell bodies are, and this is the white matter, where the axons are running through here, from the cell bodies down through the white matter. And that would conclude the brain dissection. So this is in a very short format what you need to know what's about the sheep brain dissection.